Everyone. So we're moving on again and back more related to the hazard component. So what we're going to be talking about now is something called GenRA, similar chemicals and related substances, and I'm going to explain a little bit more about what GenRA really means. So if I can get back to this one, yep, okay. And just to be a little different, then I'll pick a different chemical to hone in on. So what is this read across that we're interested in? And read across really is this data gap filling technique that's used within category and analog approaches. And there's many steps involved in developing a read across approach and performing it and in evaluating the inference that's made from it. And what we've aimed to do is really sort of been working towards developing a harmonized hybrid workflow for read across, in which we can demonstrate where there are certain components that can be more automated, or at least provide opportunities for where a more algorithmic methodology can be applied in a read across approach to ensure some reproducibility and objectivity when conducting read across. And what we've tried to do in effect is to encompass this in an approach that we call Generalized Read Across, or GenRA for short. So if our starting point in this, and what we have implemented is really a workflow within the dashboard to facilitate making these types of read across predictions. And our entry point is really sort of looking for a particular chemical of interest, and using the dashboard to sort of navigate to that. So we end up with the usual landing page in terms of a chemical of interest. And scrolling down, I've picked on dibutyl phthalate, and we scroll down to the left here where we find the GenRA beta link, which will open up a whole workflow which we can follow to make a read across prediction. And this makes it a little bit different than other aspects of the dashboard as we go through this iterative process to work through the different steps of a read across workflow in order to make that type of prediction. Hopefully this is what you end up being presented with, is this interesting looking radial plot. And in the center is our chemical of interest. So I've picked on flucosanol, and I can't pronounce things today either. And around the outside, in kind of order of decreasing similarity, are a number of different similar and similar with respect to structure analogs that perhaps will be useful in making a read across prediction. So if I hover over any given one of these analogs, you'll see a little red circle pop up with a number, and that reflects the similarity index. So when Tony was presenting similar chemicals earlier on in the dashboard in terms of based on a Tanimoto score. This is really very similar in that notion. We're providing a similarity index to show how similar hexocosanol is to flucosanol, our target chemical of interest. And if we circle around this thing, you'll see that these sort of numbers update as you hover over, and you'll see that the last one that we've picked is significantly less similar than the first one. And you can change the view, so if you want to sort of see not the 10 most similar analogs, or maybe you want to see the 5 most similar analogs, or the 2 most similar analogs, or the 15 most similar analogs, this graphic here will update, and you'll be presented with a larger or fewer number of analogs that you can focus on. Now, when you're doing a read across approach, sort of a key component, the idea there is that you want to make a prediction based on your chemical from related substances that do have some data. So we've pre-filtered based on availability of in vivo data in order to present you with not just what are similar substances, but similar substances that do have some legacy in vivo toxicity information. And that's why we have this filter here that's blocked down to automatically show you those analogs that do have data. Now here we have sort of in terms of how you might search for those analogs, there's many different ways in which you can identify things that are similar. We focused here that things that are similar based on structural characteristics or bioactivity information. So there's a number of different components here. So there's different types of structural similarity that we can use. We have here Morgan fingerprints as a default. There are torsion fingerprints, so you can search for analogs based on that characteristic, or looking at tox prints, which is another type of chemical fingerprint. Or indeed, if you're interested in looking for similar analogs based on bioactivity information like the Tox21 or the ToxCast data that Katie was talking about, then that's another way in which you can look for chemicals that are similar with respect to those types of considerations. So once you're sort of happy with the setup in terms of, I have my target chemical, maybe I don't have so much information on that, what are similar analogs that do have some information, you can move to the next sort of step of the workflow, and that will update and start to populate the next two windows in the grid-like view. So on the top here, you'll see that there's this sort of pizza tracker that sort of keeps you orientated in terms of what step of that workflow within read across you actually are in. So step 2 is really about sort of data gap analysis and generating a data matrix. So in this window, you have a kind of a summary in terms of what types of information or how many records do you have for different data streams. So for instance, in this sort of first column, we know if there is some information that's related to Tox21 information or ToxCast information in the second tab, what sort of chemical fingerprint information we've pre-generated, and to what extent are there records for in vivo toxicity information. And this one's quite a fortuitous example to focus on, because our target chemical does not appear to have any in vivo toxicity records within this database that's underpinning this tool. But whereas there are quite a few number of records for the related chemicals that we've identified in step 1. And that's designated just by this sort of simple color coding to give you an idea. There's a lot of data, and that might be designated by if there's a black coloring versus there's a little bit less data, and that's the red coloring versus sort of the lighter the color that goes, the fewer the number of records. And that's just giving you a quick snapshot in terms of how much data might exist. And moving on to the third tab, then you start to, or third window rather, you start to sort of see, well, it's one thing knowing how much data there is for a particular data stream, but you want to understand how that's spread out across different types of toxicity effects. And what we've underpinned within this approach is looking at in vivo toxicity data that's stored in the first version of ToxRefDB, a database of legacy in vivo data that was collected on a number of pesticides and records that the EPA holds. And we drilled down on aiming to make binary predictions of what sort of effect might you expect to see in target organ effects in those types of studies. So can we say whether something is likely to have a liver effect or a kidney effect, or exhibit some type of clinical signs, or have effects in adrenal glands? And you get a binary prediction in terms of, or the aim is to make a binary prediction of, are we likely to see effects in those types of scenarios. So in terms of understanding that for the experimental data that might exist for our analogs, we want to be able to have a view in terms of, well, if I'm interested in particular in making predictions for kidney effects, do those analogs that we're using have information that can inform us on that type of effect? So this type of view provides you more of a granular view in terms of not just what information exists, but how it's spread across those different study types. And you can scroll down and sort of, there's quite a number of different sort of study types, and you can scroll down this list and see these are the way in which that's sort of been articulated. Now in these different sort of windows themselves, there's sort of a bit of a navigation to tell you, what does this box in this workflow really is about, what it's doing. And if you want to hear my draining voice, then there's a little video that accompanies that sort of explains a little bit more around sort of what I'm trying to show you in each of these sort of different steps in turn. But once you're happy in terms of having a visual cue now in terms of your target chemical, what are the related similar chemicals, what is the landscape of available data that exists for those, we can move on to the next step, which is sort of generating a data matrix. And you see at the top we have an update into that pizza tracker that tells where we're now on step 3. And this view is kind of mimics the last one, but now it drills down and not telling you in terms of what data records might exist across these different effects, but what's the potency associated with those effects. And we've color-coded in blue and red. So if something's demonstrating an absence of effect, we color it blue. If there's an effect or a presence of effect, it's colored in red. So you get an idea now in terms of, well, now I've got some information about those analogs, but I can tell, you know, is there presence of effect across those target organs, across those different analogs, or is there an absence of toxicity that's seen? And if you hover over any one of those buttons or cells in this sort of grid-like view, that's sort of reflected here. So blue would say no effect, whereas if I hover over one that's red, then we sort of are presented with a number that sort of tells you what's the dose at which that effect was seen. The view here then is sort of the first chemical is your chemical of interest, your target chemical, and therefore that has a similarity index of 1 because it's similar to itself. And then in decreasing order of similarity, these other sort of source analogs and what their pairwise similarity was as reflected in that first tab. So we can start to sort of run a prediction. So now we have an idea here in terms of being able to see, you know, the available data for those source analogs, but what are their effects? And you can make some decisions in terms of whether you wish to delete or deselect one of those analogs from the prediction that you're going to be making. Maybe you decide that, well, that analog isn't really a good fit based on some other information or knowledge that you might have, or maybe you see that there's a break point in this category approach in terms of the source analogs that you've identified, that maybe it doesn't make sense to retain it in the analysis going forward. So you can make some judgments around, you know, what's the consistency and concordance of those source analogs with respect to making a read-across prediction. This is now finished processing, so now in the first column it's an update in terms of what those read-across predictions are for our target chemical of interest. And you'll notice, you know, maybe not so obvious from the screen I'm looking at, but the colors are a little washed out, right? And that is sort of reflecting the degree of confidence that we have in a given prediction that's made for a chemical. So the darker the color in this case, the less confident we are in the prediction. The lighter the color, the more confident we are. So we've used an opacity to provide a measure or a quick high-level view in terms of sort of how confident we are in a given prediction. And if I hover over, then you start to sort of see what's the overall assignment that's made in terms of a prediction that's made, and what we've used receiver operating characteristics to define the area under that curve to provide a measure of confidence, and that's also sort of reported as a metric along with a p-value metric to sort of give you an idea of how confident we are with a given sort of prediction. So in this case, the prediction, you know, doesn't look too good from those sort of statistics, but that is in part by the few number of chemicals that we're using in order to make a prediction in the first place. However, having said that, it's also sort of worth bearing in mind that this is a baseline in terms of making read-across predictions, that we're only using at this stage a chemical structure component to predict something that is quite complex in a way. Here we're sort of saying we're predicting whether there's likely to be a toxicity exhibited in a chronic study in the adrenal gland. So there's a number of other components that we haven't necessarily addressed that are important in terms of making that type of prediction, and structure alone will only go so far. So a lot of the work that we're doing going forward is considering what other components or other considerations we should reflect in terms of making a read-across prediction. And on that note, the last thing, we have a few little bugs to sort of iron out, but it's possible to download the predictions that are sort of generated and use them in other subsequent analyses, or rank order those sort of predictions to get an idea based on those performance criteria, sort of how confident are we with any given prediction and how one might rank the different toxicity effects that have been estimated and what their associated confidence are in order to move forward and make some other sort of decisions.